So we just wanted to give everybody a chance to log in and get started on this Zoom webinar. This is a webinar that we've been preparing for a couple of months now, and it's been quite an adventure. Fortunately, Myrna Tabia has been instrumental in getting this started and actually partially generating the excitement around this, which to me has come to this as a bit of a surprise. I was expecting probably hundreds of potential attendees or registrants, and the last number I just heard from our communications department, it was 3150, which is by far the biggest audience I've ever spoken to. So I would like to start off by thanking the communications department, particularly Cassidy, who's made this possible, because we basically had a very limited number of attendees possible with our standard scripts Zoom. They've actually upgraded us to the Zoom Enterprise system, which has allowed us to have a much broader population of individuals. So thank you all for attending. There's been a couple of motivations for having this. One of the motivations, not the primary one, but one of the motivations has been the release of the Activity, Metabolomics, and Mass Spectrometry book that just came out on May 1st. The book is freely available at this particular link. So if you want to download it, you can download the book. If you want a hard copy and you're in this room, you can get a hard copy outside the door. It's free. If you really want a hard copy and you prefer not to have the downloadable version, you can go to Amazon, and the royalty-free cost for the book is $13. So it's relatively cheap, but the vast majority of people opt for the free version, and you get the PDF. Everybody seems to be happy with that. Actually, one of the nice things about this has been about doing it on Amazon and also having the PDF version was I immediately got feedback on the book and a lot of corrections that I missed in putting it together. And those corrections have all been made. And even on Amazon, that has what you call a revised version from the very first version that was put on there. So that was a really nice aspect of doing this on Amazon. So in most publishing, once you have it out there and you find errors, you're pretty much stuck with those errors. So, and I plan next year to put out another free version of this with a couple of more chapters added to it in 2025. So that was one motivation. The second motivation is to give the people in my lab and the experience of being of presenting in front of what was supposed to be a moderate sized group, but this has gotten much more exciting, and I can see that the people in my lab are much more, they have a heightened level of awareness as a result of the numbers that have come in. And the third reason is that this is not going to be an ordinary short course. A typical short course goes through the very basics of the technology and then ramps up to slightly more sophisticated approaches associated with that technology. With respect to what we're going to present here today, and over the next 3 days, each Tuesday of September, is a combination of the very basics. So Corey Wang is going to talk about some very basics of mass spectrometry, but I'm going to intertwine that with some highly relevant findings that have come out of our lab recently, and that are, I think, significantly impacting the field of both metabolomics and lipidomics. And also, we're going to have a cameo speaker today at the end, Erin Baker from University of North Carolina, Chapel Hill. She'll be joining us via Zoom from North Carolina. And she's considered one of the world experts in ion mobility. So she's going to be talking about ion mobility from what she calls a deep dive perspective. So first of all, I'd just like to introduce you to Scripps Research. Scripps Research was originally called the Scripps Metabolic Clinic, and it was founded in 1924. So this is our centennial year. So it's a pretty big year for Scripps. It's been around for 100 years. They used to be located in the direction that I'm pointing in right now, about 2 miles due south, due southwest. Where they moved in the I believe it's the 1970s up to the campus where we're currently located on the Mesa. And I just thought I'd share a few pictures of scripts. This is the Eternal Flame. That was a statue that was created for Bernie Galula, one of the first dean of the graduate school here at Scripps. And behind that Eternal Flame is part of the immunology building. Here on the left is the Skaggs building, and also on the right is the Beckman building. And those buildings house a combination of molecular biologists, chemists, and a wide variety of different scientists coming from different perspectives, but largely oriented towards biological research. And this is our newest building. This is the Wong building. In fact, it's so new that we just went over there the other day to anticipate setting up a small portion of our lab for setting up within that to facilitate mass spectrometry research in the Institute. It's not even populated just yet. It's so new, but it was just completed a couple of months ago. And this is the room that we're giving the lecture in. This is we're just west or just east of the golf course, and you can see in the background a little bit, perhaps. No, it's a little bit foggy today, but behind me is also the Pacific Ocean. And I also would like to thank the sponsors. Fortunately, it wasn't a big ask. They just gave us some of the swag from AMMS. That includes Agilent and also Waters. Okay, so the course of the next 4 days, I'm going to be covering a wide variety of topics. Well, I say I, but there's a total of at least 11 of us that will be actually speaking. An introduction to mass spec and metabolomics, that'll be myself and Corey. I'm going to also talk about a topic that has gained a lot of interest in the community, and that's in-source fragmentation. And as Science Magazine has coined it, phantom metabolites. So, and of course, Aaron Baker will be talking after this about ion mobility. On the next Tuesday, we're going to have a segue into proteomics with Myrna and Elizabeth focusing on that topic. And the reason why I decided to do that was there's so much overlapping technologies between proteomics and metabolomics that I thought it would be worthwhile to kind of compare and contrast the two, at least briefly. And then following that, we're going to do have some discussions about targeted metabolomics with Elizabeth Billings and Bill Webb. And then after their talks, we're going to have population scale metabolomics by Juliana Ivanovic, who is a person at the Lausanne University, a professor at Lausanne University. And obviously, this is a highly relevant topic that a lot of people are very interested in, looking at wide populations and large numbers of metabolites all simultaneously. And then on the third day, we're going to talk about XCMS, NetLand, and a little bit more about in-source fragmentation. And a topic that I'm also very interested in, which is going to occur on that third Wednesday, is a topic that I think is incredibly unique and quite novel with respect to how many people are actually doing this right now. And and you have to listen to these words carefully because they don't usually come together very often. Absolute quantification on a subcellular level using mass spectrometry. So basically what Mike Kersey, who's going to be visiting us from AstraZeneca and as a professor at the University of Gothenburg, is going to be talking about organelle level quantification of molecules and metabolites within the organelles. It's a relatively small population of people who are doing that right now, but I believe it's one of the more compelling areas of mass spectrometry imaging right now. And then finally, at the end of the fourth week, we're going to talk a little bit about metabolite identification and finally activity metabolomics with Martin Guerre, who will be visiting from the University of Leiden. And Martin is going to discuss a 10-year-old project where he went from endogenous molecules looking for novel activity within endogenous molecules to where he is right now, moving these molecules in a modified form into the clinic. Just to give you just finalize this in terms of the speakers, Corey Wang is one of the junior members of the lab, but he basically takes care of most of the instruments in the lab, and he does all sorts of analyses across them. So even though he's one of the more junior members, he's become a very valuable addition to the lab. Elizabeth Billings is she's very talented in many different ways. Proteomics, she does a lot of protein analysis, she does a lot of small molecule analysis, a lot of quantitative analyses. And Myrna, I have to say it's largely because of her efforts that we're here today and actually able to put this on, because I would not have been able to do it myself. And Bill Webb is not only doing a large amount of quantitative analysis with Elizabeth, but also responsible for the open access labs and the advanced open access labs at Scripps. And the final two individuals who will reluctantly speak over the coming days are Winnie Urundbutai, also known as Winnie Hein. She is going to be talking a little bit about, and in actuality, she could probably fill all 4 days with the work that she's done in this area. So she actually has been building Metlin, and a lot of you probably know what Metlin is. Metlin now hosts 936,000 molecules, and with experimental MS data on each one of those molecules. And I would have to say over 95% of that data, which has been accumulated over the last 20 years as a result of Winnie's efforts. So it's really been an extraordinary effort on her part to create this. And Lin Huang is our metabolomics and proteomics specialist. I think perhaps the best way to introduce her is that she originally came from Ben List's lab when he was here at Scripps. Ben subsequently left and then got a Nobel Prize for the work that he was doing here at Scripps. And Lynn actually came with super high recommendations from Ben, and she's actually lived up to all of those recommendations. So that is basically the introduction. And the cameos, as I've already mentioned, are Aaron Baker, Michael Kersey, Juliana Ivanovic, and Martin Pierre. But you'll hear more about them later. Okay, so today I'm going to talk about activity metabolomics, give you a little bit of background on that, a little bit about mass spectrometry analysis. Next, I'm going to go into in-source fragmentation, and then finally with Aaron, the ion mobility. So the way I like to think about activity metabolomics getting started was not necessarily with mass spectrometry, but with an individual named Dilworth Woolley. So he very interestingly, he started off this area by writing a book about anti-metabolites. And the idea behind anti-metabolites was that you would find endogenous molecules, endogenous metabolites that impacted the biological system that you were interested in. That's something that could modulate phenotype. And then those molecules in themselves could be potentially used as therapeutics, or if you modified those molecules, you could then use those as inhibitors to a particular receptor or enzyme that could ultimately impact the disease. And all of you know what this is just by that description, but you typically think of it as inhibitors or drug molecules. So I'd like to think a lot of this started with Dilworth's contribution with his book, which ultimately, a couple of years later, he put many additional articles on this, including a paper in Science. Activity metabolomics, the concept is really based on the central dogma of biology. You have DNA going to RNA, going to proteins, going to metabolites, and ultimately that reflects, oftentimes the metabolites reflect the phenotype. But in reality, and again, this is nothing new, we all are very well aware of this, is that the metabolites in many ways control the DNA, RNA, and proteins. With the DNA, the control comes through signal transduction and epigenetic regulation. With respect to RNA, we all know about ribo switches and post-transcriptional modifications. And with proteins, the molecules can go in and actually cause allosteric effects, cause proteins to fold differently. They can act as substrates to the enzymes and create other molecules as a result of this. And also post-translational modifications. So when I say activity metabolomics, I'm talking about all of these types of small molecules that we observe in biological systems and how they can impact the systems and ultimately how we identify what those molecules are. And that's where mass spectrometry comes in, and that's where metabolomics comes in. So basically what we're using is metabolomics as a tool to identify the active metabolites within a system. And we'll go through this, especially on day 4 in more detail. But if you want an overview of this, in 2019, we had I was particularly proud of this review that we had on identification of bioactive metabolites using activity metabolomics. So if you want to go through this in more detail, you're welcome to go to this paper. And from the perspective of does therapeutic endogenous metabolites have any real foundation in reality? And I mean obviously I can't ask the Zoom audience this, but if I'll ask a few people in this audience right now, can you think of any endogenous molecules that exist as therapeutics? And actually silence is typically the answer I get to this. So, but you were about to say something.
Penicillin.
What's that?
Penicillin.
Penicillin, that's interesting because you could think of that as an endogenous molecule. So it's a valid, yeah. So that's a valid one. I hadn't thought of that before. I have a couple of examples here, which maybe are yeah, but yeah, the penicillin really would fall into that category as an endogenous molecule. So yeah, here are some molecules that are used. And these are typically, at the very least, billion-dollar a year molecules or hundreds of millions of dollars a year molecules. So estrogen, hydrocortisone is probably the one that most of us are familiar with because we, at some point in our lives, oftentimes you've had to use the hydrocortisone. There's a whole host of others. The one I'm particularly this anti-inflammatory. The NAC, N-acetylcysteine, is the one that I've always found fascinating personally, and melatonin is another one as a sleep aid. So again, the potential for using endogenous metabolites as themselves for therapeutic purposes is one avenue. The more common avenue that drug companies typically take is they'll take this basic structure and modify it to see if they can enhance the properties. So it's so it goes both ways, and those are typically the drugs that we see in the clinic. And from a recent discovery perspective, they're coming out on a monthly basis. There's always molecules that I'm seeing in the literature that are associated with in modulating phenotype. These are just a list of certain molecules that have come out. I think perhaps the most recent relevant one is dehydrocholesterol for cancer therapy, and that actually came out earlier this year, within I believe about a week or two of each other, in 2 Nature papers by 2 separate groups. So it's a particularly interesting molecule from the perspective of endogenous metabolites that can potentially modulate phenotype. Okay, so with that, actually, this is it. Now it's time for Corey to come up and talk a little bit about the basics of mass spectrometry, which I'm going to then segue into what I think is a really interesting area of mass spectrometry analysis.
Hi, like Gary mentioned, I will be going over a quick overview on developments within some mass spec technology and its effects on data and what we observe. So a mass spec can be easily broken down into about three major components, being the ionization source, the mass analyzer, and the ion detector. And in our opinion, the most important developments recently have been within the ionization source. So in 2002, the Nobel Prize in Chemistry is shared by two individuals for their development within ionization technology, being Koichi Tanaka for his developments with soft laser desorption technology, and John Fenn with his expansion on electrospray ionization technology. So in Koichi Tanaka's case, he helped develop soft laser desorption technology, which is commonly used in a method known as MALDI, which involves spotting your compound of interest with a matrix onto a plate and exposing that spot to a laser pulse. The matrix itself helps protect your analyte of interest from the direct energy of the laser pulse during ionization, thus minimizing the amount of in-source fragmentation caused. So the benefits of this is we can use samples in the femtomole range, but the one major disadvantage is it's not really a great method for analyzing smaller compounds due to matrix interference that comes up. In regards to John Fenn, he helped expand the research of Malcolm Dole to the use of electrospray ionization in regards to biological compounds. So the way that electrospray ionization works is I'm going to be using this can of computer duster and this front end of mass spec to demonstrate. The needle that your sample is sprayed out of is held at a voltage around 700 to 5,000 volts and sprayed directly into a spray chamber, producing a bunch of really small droplets. These small droplets are charged. And in the presence of that drying gas within the spray chamber, these droplets slowly shrink, causing an increase in charge surface density. Eventually, the forces of the surface charges exceed the surface tension of your droplet, expelling your ions of interest directly into mass spec. So in regards to ionization, what we see normally in positive mode is the protonated mass of your compound of interest. In negative mode, the deprotonated mass. And also present are some cations. For example, in positive mode, commonly you will see sodium added of your compound of interest. And in regards to ionization, Gary's going to take over in regards to in-source fragmentation.
Okay, thanks, Corey. So this particular topic, I've gotten a lot of feedback on since we originally published a paper earlier this year on in-source fragmentation and the hidden impact on metabolism and small molecule analysis. So what Corey just described was electrospray ionization. So electrospray ionization, of course, is prevalent throughout our community, both small molecule analysis for metabolomics, small molecule analysis in lipidomics. It's highly prevalent in the pharmaceutical industry. My partner works at Pfizer, and at Pfizer, they easily have a couple of hundred mass spectrometers that are routinely looking at small molecules. And typically they use electrospray ionization as the ionization approach to do these sorts of analyses. So it's again, there's a really good reason why Koichi Tanaka and John Fenn won the Nobel Prizes in 2002. And I like to think of that reason being is that they created, or at least in the case of John Fenn, it was Malcolm Dole who created electrospray ionization. John Fenn ultimately used it for biological applications, which allowed him to win the prize. And the reason for that, in my mind, is that the ionization technology of electrospray is such a soft ionization. It's a wonderfully soft ionization, such that we can even look at non-covalent interactions on a regular basis. However, and I'm going to just switch over to another technology that many of us who do mass spectrometry are very familiar with, and that's electron ionization. And electron ionization was a technology that was developed in roughly the 1950s or so. And there were other ionization approaches that were developed prior to that, but it really came to the fore in the 1950s. I actually spent a fair amount of time trying to identify exactly when it was developed, and I was actually challenged by that endeavor. I never really came up with ultimately a publication that could tell me exactly when it was developed, but it's been around at least 70 years. And electron ionization you're probably familiar with it because pretty much, I would say 99% of gas chromatography and mass spectrometry instruments have GC and have electron ionization as its ionization source. And it's so prevalent that NIST, the National Institutes of Science and Technology, over the years have created a database on mass spectral data that you get from molecules in order to help us identify what these molecules are when we use electron ionization. So here's one example. This is just a plant hormone that I randomly pulled up. And this is the mass spectrum of that plant hormone. And for those of you like me who've seen GCMS in the past, this comes as no surprise. This is a typical spectrum that you'd observe. And the thing that's really striking about this is that, and I don't want to say striking in that it's surprising. I'll say it's striking in that it's the most routine thing that you see with electron ionization, in that there's no precursor ion. Pretty much all we're observing here are in-source fragmentation. So for those of us who have been doing mass spectrometry in the last 15 or 20 years with electrospray, you would almost look at this and say, well, that looks a lot like tandem mass spectrometry. But in fact, it's not tandem mass spectrometry, it's in-source fragmentation. So in-source fragmentation has been around for over 70 years, even prior to the 1970s of electron ionization. So it's not, I got a lot of comments about our paper when it first came out and about in-source fragmentation, with people saying, yeah, we knew this all along, that in-source fragmentation actually exists. And I couldn't agree more. I mean, with respect to electrospray ionization, people have been discussing in-source fragmentation for decades. Really, it's not a big surprise. What's been really nice about electrospray ionization is that predominantly, oftentimes you'll get the intact molecular ion with the MH+. So but in-source fragmentation does exist. And what was, I think, the novel aspect of what we did was how we looked at in-source fragmentation with respect to metabolomics and lipidomics, and actually any sort of chemical analysis. And what we did, and the paper was in Nature Metabolism in 2024. I believe it came out in June or so, roughly June or July. So what we did was we took the Metlin database. So Metlin is a database that has, as I mentioned earlier, 936,000 molecules in it. And I mentioned Winnie as being the prime mover in this, but I really should have mentioned also that Ares Asporna has been, in conjunction with Winnie, responsible for the bioinformatics of actually setting this whole thing up and allowing us to actually create the database in its current form. So I really want to make sure I give proper accolades to Ares as well as Winnie for doing this, because it would not have been possible without Winnie and without Ares. So we have this database where we've collected data on 936,000 molecules. And it continues to grow. Sometimes it grows faster than others, but it continues to grow. And all of this experimental data has been generated in both positive and negative ionization mode at multiple collision energies. So it's all experimental data. There's no in silico data associated with Metlin at all. In fact, we originally did have a little bit in there, but it was so full of errors that we ultimately decided that it was more problematic with respect to helping us identify things than anything else. So we've completely eliminated in silico generated data, and everything you see in Metlin now is experimentally derived data. So, and the key to what I'm about to talk to you about is that the AMMS data was collected at 4 different collision energies. So we collected the data at 0 electron volts, 10 electron volts, 20 electron volts, and 40 electron volts. And the reason for this is that some molecules don't fragment very well, so we have to go to higher energies for some. And other molecules fragment very easily, so that's why we had the lower electron volt collision energy. So that was the rationale behind why we wanted to have a broad range of collision energy. So do we 20 years ago when we started this, do we wish we had added a few additional ones? Yes, but even with Winnie and Harry's fortitude, I don't think I can start over again. So I think we're going to stick with 0, 10, 20, 40, and that'll pretty much be it. But the really interesting one here is the 0 electron volts. So the reason why it's interesting is that, and this Chavi Domingo in my lab made this observation quite a few years ago, and we actually had a paper in Analytical Chemistry about it, is that the 0 electron volts actually mimics in-source fragmentation very well. Basically, what we're doing is we're not introducing any additional energy to the ions as they enter the collision cell. So the amount of fragmentation that is occurring is fairly minimal. So here are some examples. So here's serotonin, and this is the MSMS of serotonin at 0 electron volts. Relatively minor fragment, you can see on this. I think that's loss of NH3 or OH. Actually, I don't know. Winnie. Okay, won't put Winnie on the spot. But in any case, we actually performed experiments where we compared the two, where we had MSMS data at 0 electron volts, and we also created in-source fragmentation data on these molecules. And what you can see is that the fragmentation is a little bit more energetic with respect to in-source fragmentation. So the fragment ion, so the fragment ion there is obviously much less intense for the MSMS at 0 electron volts than it is for ISF, and that's generally what we observed. Here we have adenosine monophosphate. This is at 0 electron volts. Here we have just the in-source fragmentation. So we're seeing the same molecules. So generally speaking, the molecules that the fragmentation we observed were the same at 0 electron volts as it was for in-source fragmentation. However, with in-source fragmentation, what we typically saw was a little bit more energetic. So what I'm about to tell you, I think, is fairly conservative with respect to in-source fragmentation in terms of what we're observing in these typical experiments. Here's NAD, nicotinamide, adenine dinucleotide. Here we see these 3 fragments for NAD. Again, they're more intense with the in-source fragmentation. Sphingosine, this is probably the closest we got where they're very similar. It's just slightly more higher with the in-source fragmentation than the 0 electron volts. Okay, so again, no really big surprise. I mean, I've been told this many times that people knew that in-source fragmentation was occurring decades ago. And as with electron ionization 70 years ago and electrospray ionization decades ago when it was first came out with biological molecules, people have observed this sort of thing. What's different about what we've done is we've actually looked at it at scale. We've looked at it at in a variety of different ways. We've looked at it with respect to 930,000 molecules. So we wanted to get a sense for how prevalent in-source fragmentation is when you typically do an LC-MS run. And the best way to do this was to look at all the molecules within Metlin and see how much in-source fragmentation or how much fragmentation we were getting at 0 electron volts. So what we observed is that at the amount of fragmentation, the number of peaks that we are observing in the typical MS spectrum, which of course reflects what's going on in terms of in-source fragmentation based on what I just showed you, that the number of peaks that we're typically observing is basically 70% of the peaks are coming from in-source fragmentation. And I would venture to say that that number is actually conservative because I've already shown you that in fact there's more energetic fragmentation taking place with respect to in-source fragmentation than there was for the 0 electron volts at with MS. So just to give you a sense of why this potentially is of interest to the broader community, and I'm not, for metabolomics and lipidomics, of course, it's of interest, but it could be of interest to anybody who's doing any sort of chemical analysis. So I'll ask the question of why do you think this is of general interest to not only, so I know there's the audience is somewhat seated with people from my lab, but there's also people from outside of the lab. Any reason why you think this is of note, that there's so much of this fragmentation, so much of these peaks that we're observing in these LC-MS runs are not coming from molecular ions, but in fact, the vast majority of them are coming from in-source fragmentation? Yeah, so the answer is that in the vast majority of programs that actually look at the data that are being generated from electrospray mass spectrometry experiments or liquid chromatography mass spectrometry experiments make the assumption that these molecules are all intact molecular ions. So essentially what science made the point when they saw our article, we said, basically, these molecules are not molecular ions that are being observed in their impact, but as Derek Lowe, the author of the article, pointed out, they're phantom metabolites. They don't really exist. So why is that important? And I think that the reason why that's important is that when I say the vast majority of programs that we're looking at this are these molecular networking programs that make the assumption that all these molecules are intact molecules. And when you make that assumption, there's basically two things that you're doing. One is that you're generating these networks associated with these molecules, but you're also generating MS data on everything that's there. And when you do that, you ultimately are generating a lot of MS data that's coming from molecules that don't exist. They're just fragments, basically fragments of fragments. So in a typical molecular network that I basically randomly generated from looking at this sort of data, in reality, if you take away 70% of the peaks and make the assumption that 70% of the peaks are not real metabolites, this turns into this. And I think that obviously you can see potential issues with this. I mean, there are lots of papers out there that describe these molecular networks as telling us something that's biologically interesting and relevant. And the reality is that they just represent a lot of misannotations. So I think that the other important point to make about this is that there's been a really large field of scientists who have taken this information to mean that there's a lot of molecules that we don't know. So a lot of effort has gone into identifying molecules that don't exist. And unfortunately, it's the effort could be used in a I think ultimately a more productive way in terms of trying to come up with better ways of annotating molecules. So basically what I'm trying to say is a lot of these molecules that don't exist are in source fragments, and they are to a large extent what we commonly know as the dark metabolome. And the dark metabolome is basically the area where everybody assumed that a lot of molecules within our biological systems were molecules that we couldn't identify, but they were actually playing a significant role. And there has been a huge effort in trying to identify those. So with that, what I'm going to do, I'm going to basically discuss a little bit further. I think I've talked about it enough already, but on the third week, I'm going to present to you what I consider a simple solution to this pervasive issue of the source fragmentation and dark metabolome that I'll discuss on September 17th. So with that, Corey is actually going to transition now back to the mass spectrometer. And then after Corey discusses a little bit of the more salient issues associated with the mass spectrometer and why it's so useful for these sorts of analyses, we're going to transition to Aaron Baker and high mobility.
Hi, like Gary mentioned, I will now be going over developments within the mass analyzer and ion detector and its effects on how we perceive our data and like its results. So the mass analyzer involves a bunch of things, but basic thing is what does the mass spec actually measure? And that's going to be our mass to charge ratio. So here's an example of a peak generated from mass analyzer that's relatively older. And an important metric that we measure a mass spec on is its resolution or its resolving power. So resolution is a measure of M over delta M, which M being the M over Z of a peak and delta M being the width of a peak. So in our example of our peak generated from an older mass analyzer, its resolving power is around 200, which is relatively poor. For example, let's do an example of one after a few developments of what a resolving power 10 times it. As you can see, there's multiple peaks, and these multiple peaks are the isotopic mass of your compound of interest. So we can take this even further with another tenfold increase in our resolving power. Here's an example of that same compound on a mass analyzer with 10 and even further 10 times the increase in resolving power. So overlaid, you can see why this is important, because the purpose of this higher resolution is it can ultimately improve accuracy. Because with the example of our older peak, we take usually the mass of around the midway up the peak. And as you can see, it's slightly off of like that smaller, more prevalent peak of the example from the 20,000 resolving power mass analyzer. And as such, it's slightly off of what we're actually supposed to observe. These increases in resolving power can typically lower our mass differences to a level of less than 5 ppm error. Now, the last bit on a mass spec is our detector. So the issue with taking just the ions that come off of our ionization source and our mass analyzer is its sensitivity is relatively poor because there's just not that many ions coming off of it. So using this small bit, which is an example of our detector, we can multiply that by a factor of 1 million, thus vastly increasing our sensitivity. And this is achieved through a series of diodes of aluminum oxide diodes at increasing potentials, which inevitably cause a cascade of electrons, which gives us that 1 millionth-fold increase. And I'm going to throw it back to Gary to finish up before Aaron's lecture.
Yeah, so I'm just going to briefly describe why we've chosen mass spectrometry as the technology of choice for doing these, and that'll lead. And after a couple of minutes, we'll just jump into Aaron's talk. So, Myrna, if there's maybe you should contact Erin just to make sure she's ready. Hopefully, she's on. So, when you think of the number of molecules or peaks that you typically observe with TLC, which was used to look at these types of molecules in the past, you at most see about 50 or so. NMR is a very prevalent technology with respect to doing these sorts of analyses. However, it has limited sensitivity. What's nice about NMR is that it can be very complementary to mass spec. There are certain molecules you simply can't see with mass spectrometry that you can see with NMR, as my colleague Martin Guerra has pointed out to me multiple times. So he has within his center at the University of Leiden instruments that are designed just to look at molecules with NMR technology, and other instruments just using mass spectrometry technology. GCMS is also a powerful technology, but typically you're limited to roughly hundreds or on the order of 100 molecules at any one time. GCMS is really nice, except of course during the process of analyzing molecules, their exposed molecules need to be thermally stable because they're heated to a fairly high degree, and that has the degradation effects. LCMS allows us to see a large number more molecules when we do these sorts of experiments, and that's ultimately why we've chosen, and why a lot of labs have chosen to do LCMS-based approaches. I think the one thing that we're going to discuss now is what other orthogonal technologies can help us with respect to these metabolomic and lipidomic type of experiments. And that's where Erin Baker comes in. It's safe to say that she's probably one of the world leaders, she's one of multiple world leaders in this particular area. And I was really happy when she said that she'd be willing to present on this topic. And I love her first slide too. Great. So should I stop sharing, Myrna?
She's on. And then let me mute.
Can you guys hear me?
Yeah.
Hello, Erin.
Hey guys.
Hi. We can hear you if you'd like. You can take over.
Perfect. And you can see my slides.
Yes.
Yes.
Perfect. It's great to talk to you guys all today, and I hope everybody had a good weekend. I'm really excited to show some ion mobility information to anybody that's interested out there. So hi, everybody out in the cyberverse. So what I'm going to talk to you guys about today is what is ion mobility spectrometry, and like Gary was talking about before, how it's a complementary technique or separation technique to mass spectrometry and all the different chromatographies for the analysis of small molecules up to big molecules if you want to. The one thing that I found that's been really important when I've talked about ion mobility is explaining the difference between different types of ion mobility methods, because a lot of different vendors have different types of ion mobility. And so it's really important if you're going to buy an instrument or put one in your lab that you understand what's the differences and will some of them work the same or will some of them work differently? And then just a few applications to wrap things up. And so first, the main thing that you guys need to know is the main part behind the ion mobility concept. And that's how all ion mobility techniques, whichever they are defined by how an ion drifts through a gas under the influence of an electric field. And where ion mobility is different than mass spectrometry is if we have a tube with mass spectrometry, we want to pump that tube out of all the gases, anything in that tube with some kind of vacuum pump so that the ion will go through to the end of the tube without having any interactions with anything. We want the longest mean-free path possible so that we get a good M over Z analysis. With ion mobility, we actually want to put stuff in that tube. We want to put some kind of gas in that tube so that the ions will collide with it, but we don't want them to collide and fragment. We want them to softly collide and basically slow them down as they're moving through there and hitting a constant velocity with these soft collisions. And so a lot of different ion mobility techniques will have this electric field to pull the molecules through, because if they start colliding, they're going to want to stop. And so we want that forward force and then that frictional collision force to be equivalent, so they hit this constant velocity moving through the tube. What this allows us to do, though, is it allows us to separate molecules based on their mass, their charge, and now we get size and shape in there too. And so that size and shape lets us look at molecules that have the same mass but might have different forms. So if you have something that's open versus something that's compact, we're going to see different amounts of collisions for those different molecules with this gas in our tube. And so the different ion mobility techniques, they'll be separate and they'll define themselves distinctly by how they apply the electric field, what different pressures they use, if they have a gas flow, and then maybe if they change the temperature of their instrumental design. So I also like to show this when I talk about ion mobility, because ion mobility is not a new technique. A lot of people thought ion mobility was only from the 2000s, but ion mobility stems clear back to the late 1800s when Thomas and Rutherford were actually constructing an apparatus to study the mobility of ions in various gases. So we are clear back here. And if we think back, the father of mass spectrometry is often thought of as Thomas. So he's playing with tubes, basically, where he's pumping the gas out or actually adding gas to it. A few other dates I want to highlight in this timeline are in 1961, McDaniel actually couples ion mobility to a magnetic sector instrument. So even coupling ion mobility with mass spec is not new to the field. And then in the 1970s, there was the first available commercial ion mobility spectrometer. So even commercializing it's not new. So I always like to say that just to show people that this isn't a new technique. It's just the technologies that we're creating are really advancing its capabilities. So where people really started to get excited about ion mobility, though, was in 2006 when Waters commercialized the Synapt instrument. And this was a big deal because before then, a lot of the ion mobility mass spec instruments had to be built in-house. So only people that had the ability to build instruments could use them for their measurements. And after 2006, a lot more of these got put into laboratories where people could actually utilize the powers of ion mobility with mass spec combined. And if you look, this paper is a great paper by John McLean's group from 2015, but it shows the number of publications for ion mobility by itself, ion mobility mass spectrometry, and then different types of ion mobility. And you can see that with that commercialization in 2006, the number of publications just keeps going up and up and up. So to get at the main difference, it would be great. Like Gary said, it's more fun when you have people in the audience and you can ask, but I always like to ask if anybody knows the difference of these different techniques. So yeah, everybody always throws out the acronyms of DTIMS and TWIMS and FAIMS, but today I'm going to actually show you guys what the differences are. So when we say that I perform ion mobility measurements, it's like saying I drive a car. You can do many, many different types of measurements and use many different techniques, kind of like you can hop in a little smart car or go roll around in your fancy big Bentley. So I always like to ask people more informative questions of what type of ion mobility were you doing? What buffer gas did you use? Because that has a big difference in the type of measurements you make. What instrument do you use? What is your resolving power? Because that's really important. That tells me your ability to distinguish molecules that are very, very similar structures. What's the pressure of your instrument? And how rapid are your measurements? So you guys can memorize these if you want to sound like an ion mobility guru if somebody's starting to probe you on your ion mobility measurements. So how we start distinguishing these techniques is we're going to start with these four, and there's actually many different types of ion mobility methods. These are just the four most common that you'll come across with commercial instruments today. So there's static electric field ion mobility measurements, and we're going to split those up into drift tube ion mobility. So that's your DTIMS, and trapped ion mobility spectrometry. Then there's oscillating electric fields, which is going to be your FAMES or your field asymmetric ion mobility, and your traveling wave ion mobility. So these are the four we're going to talk about today. When I say static electric field with drift tube ion mobility, what that means is that you have a constant electric field pull across to your instrument. So here, say that we want to pull everything by 17 volts per centimeter. We're going to use a constant 17 volts per centimeter pull from the beginning of our instrument to the end of our instrument. And you can also think of that like a hill. If you're sledding down a hill, that have that constant drop as you go, and that's how you get that constant pull. With trapped ion mobility, it's a little bit different though, because we have a constant electric field, but we use different ramps of that electric field. And I'll show you guys why they do that, but you'll set it at one electric field for constant, and then we'll move to another electric field and then another electric field. So when you hear about the different ramps in trapped wave ion mobility, or sorry, trapped ion mobility, I'll show you why that occurs in just a little further, but it's also constant electric field.
So Aaron, you don't mind if I quickly ask a question?
No, that's fine.
So using these different ion mobility technologies, would you expect to see, I mean, the primary thing that you're measuring is the CCS values for each molecule, the collision cross-section values.
Yep.
Would you expect to see significant differences depending on the ion mobility instrumentation that you're using?
Nope. So you won't, and I'll show you why too as we go through this slide. So that's the cool thing is if you're in the market for different ion mobility instruments, a lot of them can do the exact same things. And just small differences in like size and the way they apply the electric field and how you get the collision cross sections. So on the oscillating electric field, though, like Gary brought up an important point is can we get the same thing? So this is where things get a little bit different, but the same. So FAMES is our kind of outlier. Is FAMES, we can't get a collision cross-section value from it. And I'll show you why when I step into it a little bit further, but traveling wave, we can. And so with FAMES, we oscillate that electric field from a high field to a low field, back up to a high field and to a low field. So the ions experience all these high and low fields. With traveling wave, though, they actually only experience the wave. So they're not feeling this high and low field. It's a nice, low, soft field that basically lets the ions surf through the device, and we can get a collision cross-section from those measurements. So the other way we split these up too is also thinking of temporal dispersion. So with temporal dispersion, it's like a race. All of the ions are eventually going to get to the detector, but they're separated by their sizes, and that's how they get to the detector. With spatial separations, though, there's going to be a slit, and only certain molecules, like maybe the center peak, are going to make it to the detector, and all of these other peaks are actually going to hit the edge of the instrument and not make it to the detector. So basically with a spatial dispersion, we can select specific ions that we want and eliminate all other ions. Where temporal dispersion, we're going to see everything eventually. And so I'll show you guys which ones are which. So here, if we start with drift tube ion mobility, and this is our nice little tube filled with buffer gas, we have an electric field moving forward, and then you're going to have the frictional force moving backward. The biggest thing with drift tube ion mobility is it's a race. So we have to have a start time and we have to have an end time. How we have the start time with drift tube is usually a square wave pulse that we let the ions in for just a specific time. And when we let these ions in, here I have two molecules with the exact same M/Z values, but one is very linear. You can see linear would basically be its definition. And the other one is packed in as ring-based structures. So we can see it's going to be a lot smaller. So it's going to move through this tube filled with buffer gas a lot faster because it's going to have fewer collisions with that buffer gas. And if I pulse those in, you can see here a nice pulse. The smaller molecule moves faster than the more extended molecule and has a shorter drift time. And this allows us to have separation where our mass spectrometer just would have said that that specific M/Z was there that corresponded to both of these ions. Now I can say by coupling with the ion mobility drift tube here that we had two different types of ions that are present under that mass spec peak. So that's where some of the power comes in. Also, you guys notice that these molecules are softly tumbling through that drift cell. So we're applying just a very, very weak electric field so that there's no dipole alignments. So what we get from this, though, because we have that constant voltage based on that buffer gas, is we get an ion neutral collision cross section. And a lot of people forget about the ion neutral part, but the neutral is very important. So the neutral corresponds to the buffer gas that the molecules are colliding with. If you change the buffer gas, it changes the collision cross section because now the molecules have different types of collisions. And so it's basically that sphere across our little molecule here. And so this value relates to the size and shape of our ion. And a lot of people say to kind of think of it as a surface area plus collisional area that the molecule has. And it's a very robust parameter, and we can compare it between my lab and labs around the world. So that's the great thing, is the drift time is going to change among different instruments, whether you have a little bit different temperature or a little bit length in your drift time, but we can take that drift time and calculate this collision cross section. And that's basically our normalizing factor so that we can do comparisons across the different labs. But do remember, your collision cross section varies depending on drift gas. So the most common drift gas used right now is nitrogen because it's cheap and it's renewable. Helium's another drift gas that used to be used a lot in ion mobility, but because it's non-renewable, people have they're not using it as commonly. So I like to always have little kind of cartoon analogies because it helps people think of concepts in different ways. And so for you guys, I'm going to do a water analogy. And so for drift tube ion mobility, basically our water here is our electric field. It's pulling our ions towards us nice and slow. So this is a nice gentle stream. And then our air is going to be our buffer gas. So the molecules are going to be hitting that buffer gas as they come along. So it's a nice low pressure system. And then I'm also going to introduce you guys to the turtles, Simon and Garfunkel. So these are our turtle twins, and they have the exact same mass. But Simon, he gets a little scared when he's in the water, and so he tucks way in where Garfunkel likes to extend out. And so these are our two different conformers for our turtles, but in the mass spectrometer, we would get the exact same peak because they have the exact same mass. So when we put Simon and Garfunkel at the top of our river here, our little stream, Simon's going to come down a lot faster because he's more compact and he has less collisions with our air gases coming through. And so in our drift tube, we're going to have Simon hitting the finish line first, or our detector first. If we move over to traveling wave though, and I'll discuss traveling wave a little bit more, we know we have that oscillating electric field. And so these waves, if we start Simon and Garfunkel from the left side, they're going to push Simon a little bit closer and closer to the finish line every time that wave hits Simon, where Garfunkel, because he's a little bit bigger, he's not going to get quite as much push as Simon. And you're going to get the same separation as Simon's going to hit our detector first before Garfunkel. So you can see that with traveling wave and drift tube ion mobility, you get the same type of shape separation between Simon and Garfunkel. And so when we go to traveling wave, the biggest part about it is with drift tube ion mobility, we can change the electric field in the drift tube to different values as long as we stay at a really soft, low electric field. If we change the electric field waves in traveling wave, our molecules are actually going to feel different forces because sometimes they're going to surf more and sometimes they're going to surf less. So usually only one specific electric field wave is used with traveling wave. And that's why we have to do a calibration. So with drift tube, we don't have to do this calibration parameter. With traveling wave, we do. And so I'm going to start our little drift tube here on the right. And we can see that our molecules are being pushed by that electric field towards the detector. And you can see that green one is actually surfing the whole way. And so the wave should probably be a little bit lowered so that it actually separates as the wave is moving to the detector. But because we need to get collision cross sections, we can actually do a really nice calibration where we take that drift time for the molecules on the x-axis, and we put it to known collision cross-section values on the y-axis, and that allows us to calculate that drift time, or sorry, that collision cross-section that we compare with our drift tube measurements and also our trapped ion mobility measurements. So basically we can get the same values out, and we know that we're getting a size-based separation in this type of ion mobility also. If we move to trapped ion mobility is an interesting technique because it uses a funnel-based device. And so I asked in one of the classes I was teaching if they knew what the funnel was. And many people didn't. And so what a funnel does is it's a bunch of lenses together that basically works like a funnel in your kitchen. It takes ions that are very widespread and tries to take them down to a small inner diameter so you can fit them through small orifices. And we can see here, we have a really nice opening here at the beginning of the funnel. And when we go to the exit of the funnel, our ions are in this much tighter orifice range. And so then we can move them into quadrupoles and octupoles and all kinds of other different mass spec components. So what the trapped ion mobility is going to do though is it has this area where you basically you go orthogonal to your funnel and you're going to spray, and then your ions are going to turn while your neutral molecules go down here and get sucked away by your vacuums. But when your ions turn, they enter this analyzer region. And in the analyzer region, there's going to be a gas that's pushing the ions towards the detector, basically. And so that's why you have an electric field that actually pushes the ions back. And that's why our electric field's actually going the opposite as drift tube here. The electric field is going to the left to hold the ions. And you get this nice separation again by the molecule sizes. But interestingly, the bigger molecules this time move closer to the detector than the smaller molecules because they're feeling more push from the gas and less hold from the electric field. And in this case, we also need to do calibration to get collision cross-section values. But I found this one is a little bit harder to explain. So Lucy Woods made this really nice animation for trapped ion mobility for me here. And so basically, here's our inlet. So this is where our ions are going to be coming from. They're going to turn, get separated in our analyzer region, and then ejected to our detector. So I'll pulse some ions in for you. So you can see the gas flows pushing the ions where it pushes the red ones, which are the biggest, further, and electric fields pushing them back doesn't hold them back quite as well. Now the little yellow ones, they're feeling less push from the gas, but more backwards push from the electric field. So now as we do those different ramps for our electric field, we're going to let the big ions out first. And so here we can see, here comes our big ions. And so those ones that exit first, so it's opposite a drift tube, where your smallest ions actually exit last based on the ramping of that electric field. And you just get a little bit different in separation, but you get the same information out of all of these three different techniques where you're looking at traveling wave, drift tube, and TIMS. We can get collision cross sections for all of those. And the collision cross section is proportional to 1 over this reduced mobility. This is your reduced mobility value. So this would basically be plotting collision cross section. But if anybody uses a TIMS analyzer, you'll see 1 over K naught on your y-intercept. So then just to finish up with our techniques, the last one I wanted to talk about, which is commonly used, is
I hope you don't mind if you go back to that last slide.
Yeah, definitely.
Just a quick question. I mean, do you, since I'm so fixated on the in-source fragmentation right now, one, you could potentially use this to get the CCS values for the in-source fragments that are being generated, but do you see much fragmentation occurring in these drift tubes? Because that would be that would make the interpretation a little more challenging.
Yeah, exactly. So yeah, that's a really, really good point. So traveling wave and trapped ion mobility have had some work done to characterize how much heating occurs in their cells. And there is a little heating because their cells do have some RF voltages to confine the ions. So there could potentially be, on some of these ones that like to fragment a lot, there could potentially be some fragments that do form, and then they would have messed up collision cross sections if they form, or if it occurred like halfway through the drift cell, it wouldn't correlate with their structure. It would correlate with actually a screwed up structure. So if you did theoretical measurements of what the structure is and you got something that was way off, you could guess that there were actually in mobility fragments that were occurring. And then the other thing is, like you said, the in-source fragments will have broken before they get to the separation region. So you would just get actually the correct structural measurement for the in-source fragments. But if things break after the separation region, then they actually break at the same time. Say this guy breaks into something smaller, so he would have maybe an M over Z that's lower. He would have a cross section that's way too high for what his M over Z actually is. And we can actually parse that out with the measurements of knowing if something broke after the drift cell, maybe in like a collision cell or something that was causing a little bit of fragmentation before we even put voltage on it.
Okay,
so it's kind of a great way to, the different structural measurements are a great way to see when things are breaking. And if you know it's an in-source fragmentation, you can circle it, but we can't deduce that from these. Yeah, and so the last one I'm going to show you guys is FAME, so the field asymmetric. FAMES, the big thing to keep in your brains is you can't get a collision cross section for FAMES, and I'll show you why. And also FAMES is also known as differential mobility spectrometry. So if you guys hear DMS, that's also the same as what I'm describing here as FAMES. And then it's also known as differential ion mobility spectrometry, or DIMS. So those three names are all pretty synonymous. It used to mean the way that the electrodes were designed, but that's kind of got scrambled throughout the last probably 20 years or so. It used to be that FAMES were planar electrodes and DMS were cylindrical electrodes, but now there's FAMES that are cylindrical and DIMSs that are planar. But you can have two different electrode designs. You can have this planar and you can have this cylindrical type of design. And what you do, and I'm going to show it with the planar because it's easier to see, is with FAMES, you have a gas flow similar to trapped ion mobility where the gas flow is pushing the ions towards the detector. And here we also have a grounded plate, and then we have a plate with a dispersion voltage on it. And so that dispersion voltage, or DV, can have either a high voltage or a low voltage on it. And when we put a high voltage or a low voltage on it, it pushes the ions in different directions. So our little blue ion, if we have a really high voltage on it, because this is a positive high voltage and a positive ion, that's going to push him down towards this other lens. Then if we go to that low voltage, he's going to feel not so much push and he's going to move up to the top lens again. And then if we do high again, he's going to go down and then go up and go down. And then because this is spatial separation, there's going to be a slit here, and this one's going to actually smash into the lens and never make it to that slit. So what FAMES uses is it uses something called a compensation voltage, and you change that compensation voltage, which alters your dispersion voltage, so you can select which specific ion you're sending to the detector. And so you put this compensation voltage on this top lens again, and then you scan through that. And as you scan through that, you're going to get these different ions out, because each time you use a different compensation voltage, they're going to feel different repulsions and attractions to that specific lens. And so in FAMES, you'll see that instead of having collision cross-section measurements, you have specific CVs that are related to every molecule. And that's the compensation voltage you put on that lens in order to measure or get that molecule to the detector. And so if we go back to our water analogy, then we have our drift tube ion mobility and our traveling wave ion mobility, which I showed you before. And then, oh, I had DMA. I didn't describe that. I should have deleted that off there. So you guys ignore that one. So if we go to trapped ion mobility, though, trapped ion mobility, I consider basically having a dam. And so the dam is basically your electric field that's holding your ions. And then you have this wind that's pushing the ions towards the dam. And so you have those bigger ones. So we have our Garfunkel is closer to the dam than Simon. So as soon as we start lowering this electric field, Garfunkel is going to go through and get to the detector a lot faster than Simon is. And then FAMES is kind of our wild child, because we have all kinds of things going on with our water. We might have waterfalls for that really high electric field we're putting on, and then maybe soft streams at the bottom for the low electric field. Then we also have the gas flow pushing the molecules. And so that's why we can't get a collision cross-section, is we don't know how Simon and Garfunkel are going to go down this waterfall. It could be Simon first, or it could be Garfunkel first. It just depends if there's dipole orientation or what kind of interactions with that high field that they're feeling. And so it's also that slit based one. And so some of them also might hit the rock based on how they're coming down this big waterfall. So that's kind of the different ion mobility methods I want to show you guys, but I really want to show you that with drift tube ion mobility, traveling wave ion mobility, and trapped ion mobility, you can get the same information. It's just 3 different instruments, and some of them have different resolving powers. And so that's where you need to talk to the vendors about how good or how different the separations might be. And so ion mobility, some of its main powers, and you guys will actually see ion mobility instruments by themselves in the airport. So if you've ever had that little swab done on your hands or your bag, they actually put that into an ion mobility instrument, and it's set to look for collision cross sections for molecules like TNT or other explosives. And if those molecules are present, that's when the little device goes off and they do further testing on you. And there are false positives. So there's times where different fragrances actually alarm, similar to TNT. But in the research lab, a lot of times we like to couple these different ones with different types of mass spectrometers, and we can couple them with quadrupoles or time of flight instruments or trapping instruments. The most common one they're coupled to, though, is time of flight instruments, and it's because ion mobility occurs mainly in millisecond separations, and the time of flight instruments occur in microsecond pulses. So that means that it's faster so that it can sample the millisecond separation. When we go to trapping instruments, a lot of times these ones occur in maybe a half second to a full second analysis. And if you have something being separated by a millisecond and then you go to something with a second separation, the time scales don't match together. And so you might only be able to fractionate something out of your millisecond separation to evaluate it with the trapping instrument. So this, and I don't know, Gary, if you're sending the slides to the students, but hopefully if the students have the slides, I just included this. This is a list of all the different ion mobility devices that are available from the different manufacturers that I mainly know about. So if anybody's interested in different ones, they can contact the manufacturers or look at more specifics. And so then just to wrap up, I just want to show you guys some benefits of ion mobility. And so one of the ones I love is isomer separation. And we can think back to our metabolites of some that are really important for glycolysis are some of our hex phosphates. And so you have your fructose 6-phosphate, our glucose 6-phosphate, and our glucose 1-phosphate. And this used to be a really big challenge for helic type of columns with chromatography because they all like to come out at about the exact same retention time. But with ion mobility, and this is probably a resolving power of about 40 in ion mobility, where a lot of these newer instruments are having a lot more higher resolving power than that. But we could get nice separation between the fructose 6-phosphate, which came out first, the glucose 6-phosphate, and the glucose 1-phosphate, all in 21 milliseconds. So a really quick analysis of some small molecule metabolites, all having the exact same M over Z, and a little bit challenging with LC. Ion mobility also helps with isotope grouping. And so our C13 ions, that C13 change, so our M+1s, our M+2s for our molecules, they don't have any structural change. And so if we have our drift time and our mass spec, we can see that we have nice isotopic envelopes for all of our ions when we look at them. And this makes informatic approaches much easier because we don't have to go back to this mass spec, which might be a little dirty with all these different ions. And we can simply look here and see if our C13 ions are at the correct abundance for what we would expect our precursor ions. Another thing that's really great is we can do multidimensional analyses. And so we can put some kind of chromatography, gas chromatography, liquid chromatography before the ion mobility separations, because many times these peaks are in the second time scale, then the ion mobility analyses are in the millisecond time scale. And then when we use time of flight, we're in the microsecond time scale. So as long as our times keep moving in shorter and shorter times, then we can get a lot of information, and we can specifically look at peaks in our LC chromatograms and see many, many other features when we look at their drift time in M over Z. So we get all of these dimensions for free when we couple these three different separations. And so each feature you detect will have information on its polarity, information on its size and structure, and then information on its mass. Another really big benefit is if people are interested in doing DIA or all ions fragmentation. With ion mobility, like Gary was talking about earlier, if the ions are fragmented after the drift cell, the fragments have the exact same drift time as their precursor ions. So if our precursor spectrum here is on the left, and we're interested in this ion that is about 780, we can look on the right and we can see all of the ions that line up below it in M over Z space are the fragments that correspond to these specific drift times and then correlate with that precursor ion. And instead of like doing DIA the hard way where you have to go and just look at the mass spec and hope that some of the fragments that you know are going to be in there, we know specifically we can slice this area and look for those specific fragments. And then one of the last ones I want to talk about is where we can actually look at the molecular packing of the molecules and compare the ion mobility and the mass spec information to learn what type of potential molecule is coming out of our analysis. And so here, if we have M over Z on the x-axis and collision cross-section on the y-axis, we can see that for specific molecules, and this is from one of John McLean's papers, that a specific M over Z, the bigger the cross section is, the more linear the molecule is, which makes a lot of sense. The more linear the molecules are, the different types of molecules that we're interested in. So if it's very packed, we have ring-based molecules like nucleotides and carbohydrates. But if the cross section is much bigger than you'd expect for the M over Z, then you're more in a lipid type of molecule. So you can learn a lot about a potential unknown feature just by looking at its M over Z versus CCS distributions. So where that's went to, though, is it's went to work in molecular databases. And so people are running as many standards as they possibly can to get this information so that we can get these class-based trends and understanding if certain molecules group together. And so John McLean's group did a lot of work with the CCS compendium, which has over 3,800 small molecule entries in it right now, with a lot of different classes of lipids, nucleosides, and organic acids. And then Gary's group, which is always fun to work with, you guys are always a little crazy. They said, well, if they can do 3,800, let's do over 28,000. So I was lucky and able to work with Gary's group to look at some of this data, which was just amazing, of how they were able to do collision cross sections on a timsTOF instrument for over 28,000 small molecules that include many, many different types of molecules. And we could plot different trend lines and learn a lot about the molecules in source and then just in mobility wise. And so these papers just came out in the last 8 months. So if people are interested in these, it should have huge potential for characterizing unknowns, but also machine learning, because we need more and more small molecule standards evaluated for machine learning. And so I hope that now you guys feel like you're not stuck under the water. Now you guys are kind of in the tunnel of water and feeling very smart with that sunshine on you. And then this is just a small summary, but I always say that if people weren't paying attention, the summary doesn't really matter anyway. So, but a huge thank you to some of the slide contributions, because I've collected these slides throughout the years to get a really good background of the ion mobility for everybody to see. And thank you all so much for your attention. And I'd be happy to answer any questions, but I'm happy to throw it back to Gary to wrap up this morning's session.
Hey, Aaron. Yeah, so thank you very much. That was excellent. I learned quite a bit from that presentation. Now, since we're right, actually, when I saw the number of slides that you had, I predicted that how long our session should go, and we're now right at the last couple of minutes. So I think I'll probably just wrap it up to say that next week we're going to be doing a little bit on proteomics, targeted metabolomics, and I think one of the multiple highlights will be Ljubljana Ivanovićević from the University of Lausanne in Switzerland. She's going to be talking about population scale metabolomics. So it's going to be another addition to Aaron's talk of a cameo that I think everybody will find highly relevant. So thanks, Aaron. That was great. Really nice.